that was exciting. So hopefully people find their way over here. What a mess. But yeah, so I can see the chat window. I can't see any people or anything else. So I guess type in slides, type in comments on the side, and I will take a look at them. So, right. So so I don't know if you can you guys see my mouse at all?
Yes.
Hey, excellent. Okay. So I have this link at the top for the supplemental, and it's going to be on the bottom of a lot of the other slides. So this is pointing to OSF, obviously, and so a lot of the code and figures as it comes later. I actually have all the supplemental online, so you'll be able to follow along, and I'll point it out later. Anyway, get going here. So this top title is a heck of a mouthful, but I did that because that's the name of the paper that we used. And the paper is actually published now online, but the preprint is also there and is the same other than formatting. And then I said the supplemental and code. So all of this work became as a methods exploration for this dual mechanisms of cognitive control, this DMCC project that I'm a part of. And for this project, Todd the aim is to be able to find analyses to be able to look for effects in individual twin pairs and heritability. So we're going to be looking for cross-session effects, across twin effects. And to be honest, when I started to get involved in this project, I was really wondering if this was going to be possible at all, actually, because to try to find the effects. So I wanted to do some methods exploration to see if I had any faith that we could get this done. But I actually now, yeah, I think that we can do this, and I'm excited to be sharing with you what the analyses are, and hopefully more of you can adapt some of these. We are bringing them further. So I'm going to attempt to convey the logic of the analysis and what we found it, why I believe it, rather than the implementation. This is all R, knitter, base R. I'm a big fan of base R graphics, so if anybody wants to geek out with me later about base R, I'd be happy to. But these supplementals are all, as you will see, knitter and base R code. So hopefully if you try to run any of them, it will work fine. Okay, so getting to it, the twins, genetic influences, that's a little bit different than what other people have talked about. But twin studies are a very classic technique. It's not anything new. It's kind of treating the twins and siblings as a natural experiment to try to see what the genetic influence might be. And this works because MZ twins, monozygotic, identical twins, share basically all of the genes and all of the prenatal, of course, because they're in the same, all the prenatal, then a lot of the upbringing, a lot of the environment. But then if you go to DZ twins, they also share a lot of the environment and prenatal, but only around half of the genes. And then if you get to non-twin siblings, they share about half of the genes as well, but less of the prenatal and environment, and then unrelated people, of course, less of everything. So that gives us a way to set up the interpretation, because if shared genetics and environment don't matter at all for the heritability of a trait, whether you have a pair of MZ twins, a pair of DZ twins, a pair of siblings, just a pair of people, the likelihood of them being similar on that trait is going to be about the same regardless of the kind of pair you made. Whereas if only genetic relatedness matters, then you have the MZ twins are much more similar than the DZ and siblings who are about the same, and then the unrelated, and then of course you can get mixes. So there's a fairly large literature on twin studies. As I said, they've been around for quite a while. There's not nearly so much for fMRI twin studies. That's, I think, mostly because of just power, that twin fMRI studies often tend to be especially underpowered. It's hard to recruit, and then you have weak, messy fMRI effects plus weak twin effects, so you need a lot of people. But the HCP data set is fairly large. So when we were trying to figure out what data set might be suitable for testing some of these methods, we went with the HCP. So HCP, most of you guys probably know about it or have at least heard about it. And it's a pretty large, there's larger ones now, data set, but around 1,200 people. And they really emphasized recruiting families, especially MZ and DZ twins plus their siblings. They tried to get as many family groups as they could. And most of the HCP was about connectivity stuff, DTI stuff, but they did do a little bit of task fMRI as well. And that's what we used. So we're just I'm only gonna be talking about the HCP working memory task. And that's the one we focused on because that is closest to the cognitive control tasks that I'm actually wanting to get the methods for. Okay, so the HCP working memory task, it's a fairly standard NBAC task, but in blocks. So there were 4 categories of pictures in the blocks: faces, body parts, tools, places, 2 working memory loads, 0 back and 2 back. So just in case you're not familiar with the NBAC thing, the way they implemented it was for the 0 back, they showed a picture at the beginning of the block, and then you had to respond every time that picture reappears. So it would be this elbowish picture in this case. But then for the 2 back, you don't have a target picture. Instead, you have to watch the series of pictures and respond if the current picture is the one that had appeared to before. So this 2 back is obviously more difficult than the 0 back. So that then gives the working memory loads. So for the purposes of methods development, we just did a subset. So we had the 2 to get a 2x2, faces and places, 0 back and 2 back. Okay, so that's almost done with this introductory part. But again, for this methods development, we really needed to have these clear load and category comparisons and predictions, because again, we needed something that we could tell if the method was working or not. So if you talk to cognitive control or psychology type people, they will say that, when you have, for the load effect, 0 back is low, 2 back is high, and we expect the strongest signal, meaning that the bold is going to vary with load in frontoparietal regions, probably some others, but frontoparietal, cognitive control-ish regions. And these regions tend to have fairly idiosyncratic brain activity. But then for category, the picture category, faces and places, we expect the that to be more in the visual areas, and we expect that to be more consistent across people. So I need some regions, and for this analysis I'm going to talk about, we just used the Gordon parcellation, and I just pulled the entire frontoparietal and visual communities, that's these 2 colors here, to define the regions of interest. If I was doing it today, if I was starting this today, I'd probably use the Schaefer parcellation. It's not that I think either of these is perfect. It's more just we needed an independent set of regions of interest, and these were handy to use. Okay, so a little bit more preliminaries, but getting closer to the methods here. So for these analysis, we use the HCP images, like I said, but we especially use the surface copes. These are so they're the results of the GLMs. Sometimes they're called beta weight images or parameter estimate images. And this is fairly typical GLM output, like I was saying. Right, so we have our 2 communities, And then, so this is surface data because that's what they had. So so I'll be referring to vertices. So for every vertex, then we have our 4 stimulus types, and then I have the 1 cope, 1 vertex for every one entry for every vertex in the community, and then these are the input for the analysis. So I'll be carrying this through the slides. Okay, so people. So I started way back when talking about MZ twins, DZ twins, siblings, and unrelated. And for the paper that's published, those analyses, these are the number of people that I included. So as most as I could, which came out to be 764 pairs. And I require, this matters more for the genetics, but obviously to have data, but then to be the same gender and within 3 years of age, even in the unrelated groups, we had that requirement. And just a month ago or so, I noticed that the HCP announced that the working memory data from some of the people shouldn't be used. And I had actually included 20 of them in the analyses that I had run. So I worked out so I actually last week for this talk figured out who those 20 people that should be dropped and dropped them, and then their twin or whoever. So these new counts are so I actually, for the analyses, the results I'm showing today are these 724 people as opposed to the original 764. And this so if you go to the website and look for the supplemental, you'll see that there's actually 2 sets of supplemental now, one with the post-publication set. Thankfully, nothing changed much, but that's definitely an advertisement for using these kinds of reports with included code, because I think it took me longer to work out which subjects needed to be excluded than it did to recreate and rerun all the analyses. Okay, so to recap this session, and if you have any questions, maybe start typing them now, and then I'll move on to more of the interesting bits. So this is and also for those of you that joined lately, so this is a 2x2 design with 4 conditions, and taken from the HCP working memory task. And it's a twin design study with 4 types of paired people. That's these MZ, DZ, siblings, and unrelateds. And I'm looking for effects of shared genetics and environment on the fMRI task activation patterns. And then the fMRI data that was released, I'm distilling out into just 2 regions of interest, the frontoparietal and the visual, and these 4 copes, as they call it, for the 4 conditions. Okay. See, is everybody cool? Maybe type something if you're all right, so I know. Hopefully you're still hearing. Good. Thank you. I was worried that something might have broke already. So, okay, so start getting into some of the more interesting stuff. Thanks. Okay, so, well, this first thing isn't super interesting, but it's always, when we're dealing with past data, it's always good to make sure that the behavior and things make sense before you move on to some of the brain activity stuff. So with the working memory, with the NBAC task, just calculated out 3 of the standard statistics that people always use with these. And there's not really a big surprise, which is good in our case, because we wanted just to make sure that everything is sensible. But performance is generally quite high, fairly close to ceiling. They got most of them right. And here I have the MZ, DZ, siblings, unrelated separately, but we wouldn't expect, for some reason to have the twins all being more accurate than unrelated people, because that's arbitrary how I split them up. But the reaction time was a little bit slower on the 2 back, accuracy a little bit lower. So that's all very sensible. So we can keep on going. And one obvious question, since these are twins, is that we might think that a pair of twins, if I'm really good at my task, then my twin might be really good at it too. And so I did a bunch. I know this is busy, but so here's person one of pair, and then, so like this would be me on this side and my twin on the other axis. And you would expect to see kind of, tight correlations if it really was the case that if I was good at it, my twin was as well. And if you look at the stats and if you kind of squint, there's a little bit of significance here, but it's not, you have to kind of squint and look at it a bit. It's not a terribly strong effect. So I was kind of like, shoot, because we were really hoping to get strong twin differences. With the heritability. So I was like, well, maybe this is just a matter of power, kind of. So I asked Deanna Barch and some other people, what is the strongest thing that you guys measured in the HCP data set that is really correlated on twins? And they said, and they suggested this little list here: total gray matter volume, NIH picture vocabulary, which is a IQ proxy, height, BMI. They said those things are really correlated. And sure enough, so here's the total cortical gray matter. And if you look a little bit, you can see this blue line. It is ridiculously highly correlated. And here we do get this pattern like what we were expecting of MZ more correlated than DZ and siblings, more correlated than MZs. So it's like, okay, it's not, so whew, we have the expected heritability pattern. So it's not that something drastically wrong happened. But it's only significant, we only get the full set of pairwise differences that we were hoping for in the total gray matter, which is the strongest. So it's like, shoot, even with this many pairs, it's still kind of marginal power, but we're keep on it going. And so this is the little teaser, and you can guess the answer that that the imaging data came out nicely. But anyway, okay, so I'm going to do just a slide or two on measuring the results in the individual people before moving on to the pairwise, even though the pairwise is kind of the neat part of it, but I think it helps to see the single subject stuff first. So as a reminder of the data, so we have these, the two communities and the four types of stimuli that we're doing. And obviously, if you have four things, you can do pairwise similarities. You can correlate all four of them together. And the usual, a common thing to do when you have 6 pairwise correlations of 4 things is you can arrange them into similarity matrices. I tend to call them similarity matrices, but it's just arranging the 6 pairwise correlations into a nice little box. And as some of you probably recognized, this is kind of akin to a representational similarity analysis, an RSA. For this particular analysis, I like to call it correlational MVPA because it's a little bit, doesn't matter. But an advantage of setting it up this way is that we can actually, before plotting these things out for the real data, we can think about, given what we know the load and similarity patterns ought to be in these 2 brain regions, can we say anything about, we can kind of put out our expectations. So what I mean by that is I've been kind of saying that if the activation patterns reflect load, then the COPEs, the fMRI images, for conditions with the same load should be more similar than those from different loads. That's kind of what I've been saying. Hopefully I didn't read it too quickly, but this is kind of like what I had written a few slides back, that if the activity patterns in whichever brain regions are reflecting loads, then like this cell here, which goes with the 2 yellow ones, the 2 0 back, ought to be more similar, a bigger correlation than these ones here, which have a mixed load. And paralleling, if they are reflecting category, then the cells that have the same picture type should be more similar. Examples from the same picture type ought to be more similar than those with different picture types. So this gives us then something, I can then plot out the data from real people and see if it looks like this. And that's what I did. And there's a whole bunch more of these in the supplemental. But these are actually for people. And I just plotted out their two brain regions here with the frontoparietal and visual. And you can tell right away that the visual ones do look a bit checkerboardy, which is good. And some of these for the frontoparietal have a bit of a darker down in this lower right corner, which is also nice. So I can, of course, print them all out for all 700 and whatever of my people and try to see if I can tell the visual and frontoparietal apart, but that's not very practical. So I need a way to quantify how well each person's matrix looks like the, trying to see this thing. So we can try to see how the, so I can try to quantify with a number to see how well it goes. So to do the quantification score, that's what these things are. So what I did here was I just took the average of the cells that I'm saying should be bigger and the average of the cells that should be smaller and do the difference. So it's a simple difference between the mean of the cells that are predicted to be similar minus the means of the other ones. This is sometimes called a contrast method of quantifying these things, but that's the summary stats I'm going to show. And it comes out just ridiculously significant. So what these are here is that each person contributes 1 point to each of the box plots. So here, this person has a really high quantification score for load and frontoparietal, but you can see that, in most people, the frontoparietal load scores are above the category, whereas in the visual, the category in most people is above the load. So it's this massive crossover effect. And well, that's what I was just saying, that obviously they have significant differences. And it's kind of nice that we're like, okay, this is another smoke test, as I think Chris said a few days ago. So it's saying that, okay, things seem to be working like they should be. Okay, so I'm going to move on now to pairwise stuff, unless somebody has another question. This pairwise is what we're trying to get to. Okay, so right, so pairwise. So that's we have these twins and siblings, and so now we're dealing with a pair of people, not just one person. So I do the same sort of thing where I have my four stimulus types, my four copes, but now I'm putting a person on each axis. So now these matrices aren't going to be symmetrical anymore, because now this cell then. So a way of making this concrete, hopefully, is that this could be if I was person one, this would be the correlation of my 0 back place cope to my twins 2 back place cope across vertices in the visual community. So I'm correlating my activity patterns with my twin or my sibling, my paired person. So this is still kind of RSA-ish and correlational MBPA, but now we're going across people. So hopefully that makes sense. So we can do the same sort of thing of trying to see what should these matrices look like given our predictions. And as a review for the single subjects, we predicted that activity patterns for conditions with the same load should be more similar to each other than those from different loads if you're in a, in our case, in the frontoparietal. But so, and then we have these symmetric matrices. And actually the same logic holds for pairwise quantification, but the interpretation gets a little bit different. So here, since it's me against my twin, you can imagine that if my brain activity in the frontoparietal and visual was totally unique, nobody else had any their brain activity had nothing like mine at all, then all of these quantification scores would be 0, because that would mean that my activity was so unique that regardless of what kind of stimulus, whether I was doing a zero back face, a zero back place, whatever, mine was totally different than my paired person. So all of the correlations in here would be more or less the same, 0-ish. So the quantification scores would also be 0-ish. But if I had like not just a twin, like a science fiction clone, where my clone was thinking exactly the same sort of thing I was thinking all through the trials, then their activity patterns would be exactly the same as me. And it would be like this sort of situation again. If I had my clone right here, it would be that, our activity patterns would be completely identical along the diagonal. So it would look like this again. So it kind of gives us a way to try to evaluate how similar the activity patterns are. So let's see, are people hearing? Is that making sense for people? Can type something. Hopefully it didn't freeze. Good, thank you. It's a little bit scary. Okay, so I got so excited to see the comments that I clicked over to the next slide. So these I just put a slot. Here are one MZ pair, DZ pair, sibling and unrelated. So these are the two matrices again, one from each brain region that we're looking at. And you can tell that it's, these are not just all blank. You can still kind of see the checkerboards, especially like here. So there's definitely some structure. It's not that my brain activity pattern is completely different than anybody else's, because if that was the case, like I said, we wouldn't expect to see any structure. But here, there is a bit of dark to the corner. It's not perfectly symmetrical. We don't have clones. So the same sort of procedure as before, where I can then quantify these. So in this case, well, I'll show the, so Roberto asked a question about averaging across sessions. In this case, well, with the HCP, I was only using the young adult, what do they call it? The regular young adult, the first HCP data set. So they only did working memory in one session, actually. And the COPEs were calculated by them. I used the HCP COPEs of both runs together. So there was only this one vector for each person. So hopefully that, okay, cool. So I have these matrices, and yeah, hold that thought, Bergolo, I'm sure I said your name wrong. Hold that thought. So now we're going to quantify these matrices again, like before. And I am going to, so brace yourself. I'm going to show you actually the full data set, because I love this figure. But it's a lot. So I'll walk you through it. How this is these are then the MZ pairs, DZ pairs, siblings and unrelated people. And I have them in columns. So like this very first column is MZ twin pair number 1 in just order by, when they're ordered by their subject IDs. And then they're also up here. So that's why the bars are not equidistant, is because there's fewer DZ twin pairs in this. Okay, so these are the quantification scores. And as you can see, it has the load and the category. So these blue L's are for the load. And if you just kind of squint at it, you can tell that in visual, these load quantification scores are all like flat at 0 pretty much. There's nothing going on. But the red ones for category, they start up high, and if you kind of squint, you can see that they sort of go downhill a bit as it goes from the MZ, DZ to the unrelated. And with frontoparietal, it's noisier, but you can see that the C's for the category quantification scores are kind of flattish, so it's noisier, whereas the L's, we have a bit of a downhill pattern, though it's not as clear. So this is the actual data set. This is the quantification scores for every pair. So obviously I can do some summary stats on them, but a lot of it you can just see in the raw data, which is why I like this figure so much. But it's clear that the load quantification scores are higher than category and frontoparietal. You can see more of the L's on top of the C's, but down in visual, it's the other way around. The categories are higher than the visual, than the loads. And of course, this is crazy significant. You almost don't need to really even try to calculate a significance. And the specificity, this blue above red and then red above blue, is present, as you can see, in all the subject groups in both communities, but the magnitude varies quite a bit between the two regions and the 4 subject groups. So if you kind of turn it around, you can do, I just did simple robust runway ANOVAs to categorize that. And you can see that the effect, there is an effective subject group where we have this higher quantified activity pattern similarity with greater relatedness, and it's most clear to see this effect in the 2 task relevant cases. What I mean by that is category and visual. So here you can see how strong the effect is. And you can, but this one, it's not much of anything. And here it's not as strong as with the category and visual, but you can still see a distinct downhill trend. And it's actually, in this, I didn't talk much about this just two back, but don't need to use the time for that. I was trying to see, Gail wants me to keep on a moving, I think. So I will try to be speedy. Okay, so I'll skip the 2 back. Oops. Okay, so this was though showing the ANOVA type plots, and then these can do the pairwise similarities for so these are the post hocs for the post hoc contrasts for the different pairwise differences, and you can see that it's with the frontoparietal we get, and these are actually stupid high significances, and so these are also suggesting that genetic relatedness. I mean, this I have to be try to be fast, but the sibling versus DZ is not remotely significantly different anywhere. So this is kind of making us think that this shared genetic relatedness is mattering a lot more than the shared environment, because it really isn't. We're not getting this DZ sibling difference. And then actually, I said these significances are so strong that it's actually, we can see these twin effects kind of more clearly in this task fMRI data than we did with straight D prime, and it's practically on the order of how strong the effects were in the total cortical gray matter, which is pretty cool, since this is task fMRI data. Okay, so just a couple of more slides. So one of them is that this genetic relatedness is explaining a lot of the variants, but there's still, of course, a lot, there's a lot more. So one obvious thing to do is, what if I sorted my pairs by behavioral performance? And when I did that, this is what came out. And I was so excited. We were still in the lab at that point. And I ran around showing all the grad students how this graph came out, because it's really nice that paired related people with higher behavioral performance, meaning D prime, so these high performers are on the right, have these higher task relevant quantification scores. So you can see a distinct uphill slope to the load, whereas with the sorting within each group, and a little bit in the visual, though not so much, but it, yeah, so it goes with behavior. And it's also with the two back, but skip that. This is just showing that, so on that plot, I was, the correlations were against rankings. It still holds if you do it with the actual D prime. So a bit of interpretation to try to finish this up. So one thing to interpret with this is that we're seeing, I think that this shows clear relatedness effects in this task bold, but it's in a task and region specific way. That if all you looked at was category in frontoparietal, so the red, or load and visual, this blue down here, you would say there isn't any effect of relatedness on the similarity. You really need to, you have to look at the category and visual, the load and frontoparietal, so both matters. And I think of that as just that doing the task is giving structure to the bold activity. And that's kind of a normal assumption. And this is kind of why we think this might be basically that to do the task well, you have to be kind of paying attention to it. And we would hope that that would make stronger activity patterns. But in this case, these stronger activity patterns in the highest performing people, they weren't just stronger to themselves, they were stronger in the same way at the vertex level as their twin was. So that's pretty cool. And this is what Bergelow was saying, that it's consistent with the idea that the visual activity is more similar between people, because even in these unrelated people, there's a lot more similarity. We still get this split. You know, it doesn't go down to zero, whereas in frontoparietal, it does go pretty much down to zero. Like, you know, the unrelated people, there just isn't much there. Maybe a teeny bit, but not remotely like down in visual. So I would say, yeah, it does show that. And that's what I just said. Okay, so to finish this off, this started with a question of, you know, are there methods sensitive and robust enough to clearly identify heritability effects in with task fMRI, even in individual twin pairs? And hopefully you agree with me that yes, this seems to be capturing some of these effects. And there's clear ways to extend this logic that I haven't talked about, that we've only just started playing with. But like, you know, I can use all these other twin pairs to kind of make or other people pairs to like make a null distribution, to compare my twin to these kind of things. There's a lot more we can do with it. And there's a lot of obvious questions and things remaining, such as what's the appropriate spatial scale to do these. I was doing these with vertices and communities. You can go down quite a bit. And we're actively running these in different data sets and hoping to see, you know, can we say something about these twins here versus these twins here across data sets, that kind of, or across tasks. We're hoping to see some of that. And obviously there's still variance beside, even after sorting people by behavioral performance. So is there something else that may describe and capture some of this variation in the quantification scores? So that is it. And I don't know quite how this works, if Gail's gonna pop up again or what? Oh, hey. I feel like I kind of sped talk through that.
No, we were here, we were listening. Thanks a lot for the talk. I would have a lot of questions because those kind of analysis are really interesting, but I fear that we've slipped behind schedule and because people
Yeah,
might have.
well, but it started
Uh,
like half an hour.
I know it was really hard to get this started. I am so sorry, Joe. Thanks a lot for this talk. I would so much want to discuss this.
Yeah,
Uh,
well,
you know what? Here is a
definitely
suggestion.
anybody, yeah, if anybody wants to email me or whatever afterwards, let me know because this stuff's cool. So,
Here's a suggestion. Let's hear Tor, and after we can move to the online town if people want to stick around, and we can have a discussion if people are still around or by email. But because of the time
sounds good.
constraints, everybody has a schedule. I think we have to move to Tor.